Hi and welcome to today's presentation from BioStock Studio. The presentation is from Biosurgeon and CEO Peder Andersen. Welcome Peder, and please take it away.
Well, thank you very much for the invitation to present you some facts of our company and our project. The company name is Biosergen, and we are a company that is developing new products within the antifungal sheet. And it is our mission to make more products available in a high medical need area where it can help thousands of patients every year. We do have one of the big unmet medical needs that is haunting the antibiotics and the antifungal space. There is an increasing health crisis due to resistance development with the current medical products. So what we are looking after is patients who are getting infected either by opportunistic infections, either hospital-acquired infections, or anything that is coming out in the countryside or in the cities with patients who have a low immune resistance. The majority of these fungal infections are actually coming from a few strains of fungus. It's called Candida, it's called Aspergillus, Cryptococcus, and Pneumocystis. And it is the emerging threat is coming from multi-resistant and resistant fungal strains, fungal pathogens. And Big Pharma has for the last years pulled out, or for more than 10 years actually, pulled out from research within this area. And therefore it is from the organizations being considered a health threat. This is our pipeline, and we expect that we can have the first product submitted by end of 25 with an approval in 26. BSG-005 is now emerging into the clinical phase 1, which is one of our goals, and then we will continue phase 2 and later on phase 3. We also have 2 other products, formulations in development, both looking at the same molecule, but with a nano formulation that is lung targeted and one that is oral. These are important new developments that we have put in place as well. BSG-005 is a unique antifungal. It is because we have done a terribly lot of research to isolate this molecule and have it becoming stable and easy to deal with. We have done that with the help of the Research Council in Norway, and we have used all the high technology methods that we could actually get onto this one. And we have basically modified several gene clusters in the host organism, which is a bacteria, to actually produce this molecule. It's called a polyene. And polyenes are fungicidal, which is a major difference from all the others. They are known for their very low resistance development. Amphotericin B is the oldest product here, has been on the market for more than 50 years, and its effect is undisputed, and the resistance building is virtually non-existing. It has, however, been used as a last line of defense because it has a toxic effect on the kidneys. Contrary, for BSG-005, our data shows us that we have no nephrotoxicity, which is a major step forward for this group of products. And we have done all the research for over a number of years, and we have published in more than 20 scientific publications, and it has been really reviewed and peer-reviewed publications all the way. So the basic research is good. Here are some of our in vitro test results that we have on two of the most normal fungal strains. It's Candida and it's Aspergillus. As you may see, the Aspergillus is a difficult strain, and what we see is that most of the other products are not able to kill the bug. Only BSG-005 and to some extent one other product, including Amphotericin B, is working on these Aspergillus resistant strains. Here are some in vivo data that we have in our mouse model, a very well-known model that is being used for almost many years, but also has the majority of all testing in this field. So we have done it against Candida and we have done it against aspergillosis. And the data shows us survival rates. And what we have done here is we have tried to test equal doses of Amphotericin B and BSG-005. And what we can see is on the left-hand panel that we have a very high survival rate, 80 to 90%, compared to Amphotericin B, which is lower here. So on a basis that looks at similar dose levels, we have a higher potency. Similar on the right-hand slide, what we see is that we have a slightly lower, because Aspergillus is a difficult one, we have a slightly lower one, but we're still ahead of Amphotericin B ambisome, even though we are at a dose that is relatively low. Just to compare the toxicology in mice that we have done, and in the acute studies that we did, we have a maximum tolerated dose that are 4 to 5 times higher than what we see with Amphotericin B, and similar on the lethal dose, meaning where 50% of all animals are dying, we can see that we have a much better tolerability on that level. So it's a 5 to 6 times higher. One of the issues are around the kidneys. And here we have on the right-hand panel a number of test data showing the NAD, which is an enzyme marker of early renal tubulary injury. And what we see is that there is almost a linear connection for BSG-005, whereas Amphotericin B is just increasing, even at low doses. And this is a major difference from the current products in this polyene group compared to BSG-005. So that's what we're looking for, a product with a higher potency and with a better safety, and specifically more or less nothing on the kidneys. So that's our goal. Here we have a clinical program that is designed to give us a filing date by the end of 2025. And what we are doing right now is having our phase 1 coming up to run in Australia. And we will do that in healthy volunteers to investigate the safety and the tolerability. Then we will look into a phase 2 program with several trials, and we will try to fully look at the indication profile for BSG-005. And then we will get into phase 3, but that depends, of course, on both our data, but also the discussions with the FDA, whether we will need one or several studies, depending on the data. Other important matters is orphan drug status. And we have applied for that with FDA and with EMA, and we will get either approval or response within the next few months for this one. And we will likely also be awarded GAIN status. GAIN is an incentive program from the FDA because there has been so few new drugs within the field of antibiotics and antifungals. So that's where we are, and that's our immediate goals for 21 and early 22. Thank you.
Thank you for the presentation. And I have a couple of questions for you. Like you mentioned here, there have been very few, if any, drugs developed in this area in recent years. How come?
Well, Big Pharma has withdrawn from this area over the past 10 or 15 years because it can be very expensive to develop new drugs within the area. And also, they have other interests. Oncology has been very high, neurology has been very high, and some of these are also a little bit more profitable because antibiotics and antifungals are short-term treatments. Yeah. So that's some of the main reasons why.
And if we turn to look a little bit more at Biosurgeon and what's happening with your company, you are now listing the company. How come you have chosen right now to do that?
We have, over the past many years, had a number of very loyal and supportive investors and investor groups. But going into the clinical phase is simply too expensive for our investors to continue giving the support. The cash and the need for cash is so much higher when you hit the clinical phase.
And if we then look for the rest of the year, what are the most important milestones for Biosurgeon during 2021, other than the listing, of course?
For 2021, we want to have our orphan drug status approval from the FDA and the EMA. We want to have our phase 1 trial up and running in Australia. And we would like to, or not would like to, we will have our manufacture for phase 2 done or started so that we are ready to enter into phase 2 mid next year.
Well, thank you, Peder, so much for coming here and taking the time to tell us about the company.
Thank you for you.